So first of all, thank you, Danielle and Carol, for this wonderful meeting. It's a great pleasure to be here. I'm Letusa, and he's Leonardo, and we are conducting this beautiful section of malaria pathogenesis. So we have in this session three great speakers: Fábio Trindade Maranhão Costa from Unicamp, Matias Marti from Switzerland, and Sylvie Garcia from France. We are going to start it now with Fábio. Fábio is a full professor at Unicamp. He's working with immunology and host patho interactions for more than 20 years now. He's a researcher, as I said, in Campinas, and he's also focused in potential therapeutic strategies. Last year, Fábio was honored with the highest award in Unicamp University, the Severino Vaz Award, what is actually a really great achieve in his life and in his career. And myself, I'm very honored to introduce him today because he's not only a great researcher, but for me, he's a great mentor as well, as for many of people, many of the students and people that are here as well. Fábio, welcome.
Hi, good morning. Gosh, last night was intense. So first of all, thank everyone to be here. Really nice night last night. And I also like to thank Danielle and Carolina for this wonderful meeting. This has been so great, so great, so great. Thanks a lot. Okay. So many years ago, that was the Jurassic era of malaria. I started to get interested how virus could complicate some cases of Vivax. So I tried to understand what was behind these complication cases of malaria, Vivax malaria. So back there, that's long time ago. So George was in this paper too, Laurent. That was one of my first papers on my lab. We just realized that P-Vivax could, P-Vivax could cytodere in the endothelium cells. So asexual blood forms could cytodere in the endothelial cells. And also, those gametophytes could cytodere in the endothelial cells. So I thought, okay, that might explain why Vivax could complicate in some cases of the infections. So I started to try to understand this. We move, oops. And with Letusa, we did this work, and then we started to take a look on the rosette forms of Vivax. So Vivax as falciparum, they do the rosette, but by that time we realized the rosette process in Vivax is quite different from the falciparum. Although the rosetting process was not involved in complications. The rosette process was involved much more in downregulation the phagocytosis in the host. So by that time, I started to realize that although Vivax has quite similarities to falciparum, the pathological process is completely different. So I started to try to go further and further on the pathogens of Vivax. So then, with João Luiz and João Conrado, João Luiz was back there was a postdoc in my lab, and João Conrado MD, PhD program. We select almost 100 P-Vivax patients, and we try to understand what's going on, what's under the surface when you take a look on the clinical and immunological aspect of those patients. So in the beginning, all those patients, they looks like pretty much any malaria, Vivax malaria cases with loss of thrombocytopenia, loss of leukopenia, pretty much what you see in the Vivax case in Manaus, in the Amazon. But when we started to take a look in several immunological and erythropoietic molecules on those patients. We just realized when we do this cluster analysis that you pretty much have two types of, let's say, profile of those patients. One that you see here in red that you call Vivax high. So those patients, they have a high modulation on these parameters, immunological and erythropoietic parameters. And another class, the blue one, that you call Vivax low. They have really not much modifications on the immunological and erythropoietic parameters. And the funny thing about this, the peristemia level of those patients does not differ that much. And what was different on the patients was pretty much the biomass of those patients. So by that time, we started to realize that the peristemia was not that relevant for those high modulations, immunological and erythropoietic modulation of the patients, but the biomass was quite important, was really key factor on those patients. So I started to think, okay, peristemia levels is not that significant, it's not that the major thing. The biomass is the major things. But the thing is, the peristemia, as you can see here, does not correlate with the biomass. So the question is, where is this parasite? If this parasite is not the peripheral blood, where this parasite is hiding? So that became, in my lab, the most important question that we have made the Vivax for the last years. Where is this parasite? And what this parasite is doing somewhere else from the peripheral blood. So that's a biopsy that Marcus have. They have this collection for the series of, I believe, 17 biopsies from patients that die with direct with Vivax or indirect with Vivax malaria. That back 15 years ago, John showed those data, the spatial transcriptomics yesterday. So that's a lung. So Max collect this, and when you see this Vivax here, but not much. So the amount of Vivax in the lung does not explain at all the complications of that this patient did have. So what's going on? So, okay, Vivax has not much in the peripheral blood. They have this huge biomass, but where is this parasite? It's not in the lung. Okay, so let's continue studying. So that's when came out the studies of Matthias with Nicanor, with the primate, non-human primate, showing lots of gametophytes and schizonts in the bone marrow. So, okay, so this parasite, they're in the bone marrow. But those primates, these primates, they have no spleen, right? So we're going to take a look on this meta-analysis that we did published in Trends. We see that the bone marrow and the spleen, they harbor the huge amount of P. Vivax. So back there, we start to take a look, okay, let's try to go further and further and try to understand what's going on in these two major reservoirs, the bone marrow and the spleen. Other thing was really important. Vivax was not passing through the bone marrow, was not just passing through the spleen. They could really damage the bone marrow, and that was Marcelo Brito that presented yesterday, and Marcelo showed this Journal of Activities paper. We have a huge erythropoiesis, the peristemia is normal, and you have anemia on those patients. So Vivax inside the bone marrow could really disrupt the bone marrow. So it was not just passing by, it's doing really something really damaging in the bone marrow. So that's when Matthias called me, that's what from Booth here in Brazil, and said, Fabio, we'll have to start a new project, very ambitious. So let's try to collect bone marrow from patients, so blood bone marrow aspirates, peripheral blood. Let's go for transcriptomic, single cell, RNA book, immunological status, everything. Let's be bold on this project. And I said, okay, Matthias, this is just great, but it will be quite complicated to do this. Took us maybe 1 year and a half to set up every conditions, and we done. We have the help to Gisele Manaus, Marcos, Eric is a hematologist from Unicamp, because back there, to establish all the protocols, we had to have some bone marrows, not for infected patients. So if this collaboration with the blood bank, all the patients that they have the leukemia, they have to come back and donate the bone marrow to be checked. So when this patient were in the last visit for the bone marrow checkup, we take the bone marrow aspirates and try to establish all those protocols that you're going to see. So it took a lot of time. So Anne Marcel also helped a lot. Jessica that presented 2 days ago, she's key in this project, Camila, Letuz, and João. So have this fantastic team to go, and that's a new project that's going on in my lab, and Matthias' lab, and Gisele's. Okay, so this was one of the, I believe, key data from the bone marrow that you had, and that I started to think how deep the bone marrow has been modulated on those patients. So can you see here, we have the uninfected patient, the bone marrow acute malaria, peripheral blood acute convalescent malaria. And when you see 42 days after the day 1, so they get treated, they have no more malaria, we get another bone marrow aspirate by 42 days after the acute infection, and the bone marrow is still modulated. So this parasite, what he does in the bone marrow has a profound consequence for the host. So really start to think, okay, let's go deeper. Let's try to understand what's going on. Another thing quite interesting when you do this PCA and clusterization analysis, the bone marrow convalescence parameter do not clusterize with the health donor parameters. It seems that it's being high modulated and it still shifts by 42 days after the infection. Okay, so we established the first single cell for bone marrow aspirates, and that is from the host, so all the lymphoblastic, all the cells from the host. And what we saw was, first of all, so you have the peripheral blood, bone marrow, the profile is different, and you have high proportion of platelets in patients in the peripheral blood, less the health donors. And also, that was a really major discovery, a decrease in B cells on day 1, an increase of B cells in day 60. So really shifted the B cells production in the bone marrow. And also in the bone marrow, that was quite interesting data. We have a high number of erythroid precursors and a reduction on the late erythrocytes. So the parasite inside of the bone marrow really modulates the myeloid cells and really modulates the erythropoiesis. So, what do you see also on those data? First of all, we have, when you take a look at the morphological smears of the blood, these aspirates, we see that erythroid maturation defects, because you have this bridge between erythroblasts, so really marker for erythroid maturation defects. And also you see a huge amount, increased erythropoiesis in the early infection, and also the erythroblast and bone marrow show an increased proportion day 1 compared to day 60. So we really have a shift in the bone marrow. So we increase erythropoiesis and we stop the late erythropoiesis. So really the parasite is really disrupting the bone marrow of this patient. So when you take a look on several molecules from the ear erythropoiesis and the terminal erythropoiesis, we really see that the increase of erythropoiesis at the early infection stage and the reduction in the rate erythropoiesis. So really pushing the host to keep going on the early erythropoiesis. So pretty much that's a hypothesis, forcing the patient to produce more and more reticulocytes, early reticulocytes full of CD71. And also you see erythroblasts in the bone marrow showing increased proportion day 1 compared to day 60. So the parasite inside of this bone marrow really shift what should be there. Also, we see that PVEF infection disrupt the bone marrow B cell differentiation. So what you can see on this data, we have loss of increased production of plasma cells and the decreasing production of premature B cells. So they push the plasma cells to produce loss of antibodies and they modulate the B cell maturation at the same time. So really modulating what's going on in the bone marrow. So we have a decrease in pre-B, translational B, and naive B cells, and at the same time, expansion of plasma cells in day 1, and also at day 60 after infection. So this process goes from day 1 up to day 60. It's amazing. And also you can see in also these dot plots, we have lots of expression of key genes involved in B cell activation, like ETC, BCR, B cell receptor, is quite different in the bone marrow for health and infect patient. And also you see an increased expression of specific genes in plasma cells, even at day 60. So we really have a shift in the erythropoiesis and the myelopoiesis of those patients. We also have production of antibody response that's not different from peripheral blood from the bone marrow. So still have antibody in the bone marrow and antibody in the peripheral blood. The response pretty much in the same for all antigens. So if you try to find a marker for the presence of the parasite in the bone marrow, it's not going to be that easy because you have a quite similar antibody response in these two compartments. So, okay, we said, okay, let's see now the parasite side. Pretty much I was talking about the host side of the infection. So the B cells, the erythropoiesis. So let's see what's going on with this parasite. Okay, so Jessica established all this protocol to separate off all the CD71 positive cells. We enrich these to do the bulk RNA-seq and also the single cell. So that was the first and quite interesting data that you have. That is bulk RNA-seq. And although we have the same Vivax everywhere, right? It's the same patient. So you compare blood marrow and the peripheral blood. The transcript in the bone marrow is quite different from the transcript from the peripheral blood. In the bone marrow, you have much more transcript involved in invasion, much more transcript involved in sexual commitment. So the question is, the parasites pretty much they have different phenotypes, different personalities. So the question is, which one is the parasite in the blood? Is the naive one? Which one is the phenotype in the bone marrow? Is the aggressive one? And which phenotype is in the spleen? Is the correct one? Is the smart one? We have absolutely no idea so far. The thing is, although you have Vivax everywhere, depending on the compartment, spleen, bone marrow, peripheral blood, they're different, they have different personalities. And maybe this can have different consequence on the fate of this patient. So when we start to do some functional assays with this bone marrow and peripheral blood parasites. And what we see, as Jessica showed this yesterday, the bone marrow samples we have when you do these mosquito feeding assays, the bone marrow really will have the low infection, will have a lower number of OOCs compared to peripheral blood. So pretty much confirm, validate that data that the transcript is from bone marrow is different from the transcript from the peripheral blood. So we start to validate those data. And, okay, sorry. And the other thing that you saw, that's rosette production. So you see that in bone marrow, we have high numbers of rosetting, and also have the rosette in the bone marrow are larger than the peripheral blood. But what's doing this? We have so far, we don't understand why we have this difference, the rosette, the size of the rosette, and also the number of the rosettes in the bone marrow vessel in the blood, in the peripheral blood. But in the bone marrow, we have lots of rosettes, and the rosettes are much larger than the rosette in the peripheral blood. And then, this is just a preliminary data. We tried to create the first atlas from the single cell bone marrow and the peripheral blood. So far, this is the first data that you have. Our data really looks like this paper published by Sa in 2020. That's primates. That's not the ideal gold standard for comparison. Could be much better if you have like a individual infected with vivax with larger sample. I believe this is 8 primates, and here we have just 2 patients. So we still have to come back to the endemic area, get more samples, but so far we were able to create this first assets from bone marrow single cell. And that's the last data that João is working hard for these last years. This is the spatial transcriptomics. So that's a biopsy from the bone marrow. And we can see these lithobrass islands is that when you see the distance from the cells from the macrophage, normally when you maturate the reticulocytes, you can increase the distance. And from when you have this infection with vivax, we don't have this, the distance is still pretty much the same. So pretty much showing that in the bone marrow, the infection really disrupts the architecture of the bone marrow. So the modulation of the bone marrow by vivax is really huge and could really disrupt the architecture of the bone marrow. So I'd like to thank all my team that has been passed from my lab from the last years, that are fundings, FAPESP, Gates Foundation, VR for Switch and MRC, and that's the Instagram from my lab. And that's all my collaborators, especially in Amazon, Gisele, Marcos, everyone, and all these universities that participate. Thanks a lot.
Thank you, Fabio. I'd like to invite Dr. Matthias Mark. Cheers now. So, Matthias is a well-known presence in the malaria meetings here in Brazil in the past years, so it's a great pleasure to have you back. Matthias got his PhD at the Institute of Parasitology in Zurich. Then he went for a postdoc with Professor Alan Kalman in Australia. And then he moved as an assistant professor in Harvard for a few years. And then he was hired as a professor in the University of Glasgow. And I believe since 2022, he went back to his homeland, the Institute of Parasitology in Zurich, where he works as head of the Institute of Parasitology. So it's a pleasure to have you here, Matthias.
Can you hear me? Thank you very much, and thank you very much also to Coral and Daniel and Fabio for the kind invitation to this meeting. It's exciting to be here. It's now the third malaria meeting in Brazil that I'm attending, and I'm really impressed always by the fantastic science that you guys are doing, but also maybe even more so by the collaborative and inclusive spirit of the malaria research community here in Brazil. I think in Europe we could learn a lot from you. So it's really great to be back, and I'm very happy to be in this session here. Okay, I'm just going to use it. Yeah, I can use it. Yeah, okay. So, as was mentioned briefly, I'm in Zurich at the moment, but actually I'm sharing labs between Zurich and Glasgow, and so we are somehow an integrated team. And this is just to mention briefly, what we are doing in the lab is usually we are developing tools, and then we are using these tools to define phenotypes, and then with these phenotypes, eventually we want to go to mechanism as is shown here, and eventually to translation. And so this is what we have been doing with several aspects of malaria biology, in particular with regards to transmission. And today, of course, I will focus on our work on Plasmodium infection in the hematopoietic niche. So this started when I became an assistant professor at Harvard in 2007, and at this time a colleague, Danny Milner, an excellent pathologist, worked at the Brigham and Women's Hospital, and we were bouncing ideas for my first big NIH grant, and my hypothesis that I had was that the gametocyte would be adhering somewhere in a tissue or in an organ, much like the asexual parasite. And so he introduced me to his colleague, Terry Taylor, who has had an ongoing autopsy case cohort in Malawi. And Terry kindly invited me to join this study. And so at that point, then, we wrote an NIH grant and eventually got it. And I got my first PhD student, Regina Choice, and we embarked in this large-scale study where essentially we selected 20 patients that were HIV negative, had a short post-mortem interval, had detectable parasitemia, and we were able to do immunohistochemistry based on the quality of the tissue. We then quantified parasite distribution across different tissues using H&E and stage-specific antibodies. So I'm not going to go into that study because it's already more than 10 years old, but essentially this study and then several, a parallel one that was led by Alfredo Mayor, and then subsequent studies that we and others did, established this emerging paradigm of parasite infection in the hematopoietic niche. And so the important note about that is that it's not just in the vasculature or in the sinus, in the sinusoids of the bone marrow, but it's also in the extravascular environment that you can see here. So this extravascular environment, as Fabio and others already noted, is where erythropoiesis occurs, where the young red blood cells are, which also falciparum preferentially invades, and which reticulocytes, which Vivax exclusively invades. So this was very exciting, and we could also find parasites directly in these erythroblast islands where erythropoiesis occurs, and it posed a lot of questions, of course. So how do the parasites interact there? How do they actually enter this space? How do they exit? How conserved is this phenotype across different species? What does it mean for the host? So we heard a few things just now from Fabio about it already. So we were able to recapitulate this finding in Vivax, as you have seen. We were also able to recapitulate it in the rodent malaria model. Importantly there, the spleen is also hematopoietically active in adult animals, and so therefore the spleen there played a much greater role. And we were also able to do intravital imaging in the mouse model, where we were able to show how the mature gametocytes directly passed through the endothelial slits to basically get back into circulation in preparation for mosquito transmission. So this concept evolved over time, but at the same time, our colleagues Nick Anstey and Stephen Coe embarked in a pretty incredible cohort study in West Papua. So Nick Anstey and Stephen Coe. Stephen is a clinician, and Nick is also actually an MD and public health expert. So they're at the Menzies School in Darwin, and they embarked in this study in West Papua where both falciparum and vivax prevalence is pretty high, and you have a lot of motorcycle accidents. In particular with male adults. So the combination of the two, they thought, would maybe give them an opportunity to look at the spleen from motorcycle accidents where these adults had splenic infarcts, which in fact that happened. So they published a study. We were just collaborators because we did some histology with them, but essentially they had 9 falciparum infected chronically infected adults with splenectomies and 6 vivax, and then a whole bunch of mixed ones. In some of them, they couldn't find any parasites in the blood, but all of them had parasites in the spleen. And so what they were able to show is that parasites were in the spleen, in the sinusoids, in the cords, in particular, and many of these parasites were actually not in macrophages, because they did some staining with that. They were basically probably viable parasites in the spleen. They had spleen and blood, so they tried to calculate how many parasites are in the spleen compared to the whole body. So for the blood, they just extrapolated the blood volume and had the parasitemia, so this gives them a total volume of parasites in the blood. For the spleen, they also quantified the parasitemia across sections and then extrapolated the spleen weight. And so they came up with a proportion of parasites in spleen versus blood, which gave them the incredible numbers like 99 nearly % of all parasites of vivax in the spleen compared to blood, and about 93 in falciparum compared to blood. So this and then in addition, you see on the bottom right there, they were also able to show that in the spleen were most of the parasites in CD71 high reticulocytes, which are the youngest ones that have just emerged from the bone marrow. And so all of this together established another new paradigm with these 2 companion studies in the New England Journal and PLOS Medicine, suggesting that the spleen is actually the major reservoir for parasite infection in in falciparum and vivax. It carries or it facilitates an endosplenic asexual cycle. It is also a reservoir for gametocyte production, and it harbors a huge reservoir of reticulocytes. So this was very exciting, but also a bit controversial because it hasn't been shown before. And of course, for us, it also posed the question, well, did we just miss that in the autopsy cohort in Malawi, or is this something that we should probably have another look at? Which then, of course, we did. So I had another PhD student, Lauren Galloway, at that time now in Glasgow, and we were still collaborating with Terry and Carl and the team, and in the meantime also with Chris Moxon, who has a joint appointment in Glasgow and in Blantyre. And so we had a series of questions. Is there a significant accumulation of non-phagocytosed parasites in the spleen in this pediatric severe malaria cohort in Malawi? So this is a cohort where autopsies were performed between 1995 and 2010, in total 103 fatal cases of children. Is there evidence of an endosplenic cycle? Is the spleen biomass significantly higher than that of other tissues? And does the spleen contribute to gametocyte formation and development? So, of course, the big advantage for us was that we had an inventory of tissues across the whole body, so we could actually compare. And so the first thing we did was we didn't want to go through all tissues again, but we wanted to specifically compare spleen and bone marrow. And then we also used the lung as a vascularized organ where we actually could get an estimate of peripheral parasitemia, because these autopsies, we don't have blood samples. And so the first thing is we basically calculated the peripheral parasitemia based on the parasitemia in lung blood vessels using CD31 as a marker for endothelial cells and HSP70 as a parasite marker. And then we just looked at the parasitemia in spleen, bone marrow, lung, and the lung large vessels, and compared with the co-study. And you can see, so the parasitemia indeed is highest in the spleen with about 2%. That's the mean from the same 20 patients that we also analyzed previously in the original study, which was similar to what Stephen Coe saw in these chronically infected adults in West Papua. But then, when we looked at the biomass and did the same, so we basically extrapolated the spleen to the spleen weight, the bone marrow tissue from the whole body, the lung, and then the peripheral blood parasitemia, you could see that actually the biomass across organs that we analyzed is not different, but it's of course higher in all these organs compared to the peripheral blood. So this was clearly very different to what Stephen Coe and colleagues saw in West Papua. And the splenic parasite retention rate, which is basically the retention of rings that is known to be about 10% through the splenic filtration, was also about in the range. So there's not a bigger retention in the spleen that we could calculate. And so then the next question was, is there significantly more is there an endosplenic cycle in the spleen? And so the way that we, and also in the previous study by Coe et al., it was calculated is, so the falciparum cycle is 48 hours. Rings and early trophs make about 50% of that, and then trophs and schizons make about 25 each. So we should find the same proportions in the spleen in these compartments if there is a true endosplenic cycle. But we didn't actually. So most of the parasites across the compartments, so this is in particular the sinusoids and the cords, were rings. There's very few trophozoites and schizons. The early trophozoites and the early gametocytes we couldn't differentiate by GMSA, so these we just put in as unclassified. But essentially, the stage distribution did not support the presence of an endosplenic cycle, but the distribution of the parasites across the compartments was similar to what was already published previously by our colleagues, Coe et al. We also didn't detect any gametocytes, suggesting that there's not really an accumulation of gametocytes in the spleen of this pediatric cohort. So then, of course, another major question was, so is there gametocyte formation going on? And so to further get at that, because we couldn't classify them by GMSA, we went back and did some H&Ds using PFS16 as in the original study, and then we also used an additional antibody for a merozoite surface antigen, which we have separately validated as a marker for sexually committed schizons. And the idea was, if we could find these in the spleen, that will support the formation of gametocytes in the spleen. So we could find PFS16 positive cells, as is shown there in B, mostly in the red pulp, of course, but at a very low parasitemia, below 0.1%, so about 0.05. And we couldn't find any MSRP1 positive cells. So this is very different to what we originally published in the bone marrow, where we found extravascularly about 50% of all the parasites to be gametocytes. So in the contrast then, in the bone marrow, of course, we find PFS16 positive parasites in the parenchyma and in the sinusoids, and also we find MSRP1 positive parasites there. And if you compare, you can see that at the bottom here, so in green is PFS16 and in red is MSRP1. So the proportion of PFS16 and MSRP1 positive cells is similar, suggesting that really gametocyte formation occurs in the bone marrow parenchyma, leading to gametocytes because we could directly now quantify that across multiple patients in the bone marrow. So with these data then, we went back to our questions. So is there a significant accumulation of non-phagocytosed parasites in the spleen in acute pediatric P. falciparum? Yes, we see them accumulated in the spleen, but no. Then the second question would be, is there an endosplenic cycle? We can see schizons, but it's not compatible at a rate which would support or suggest an endosplenic cycle. And then we see significant differences of the splenic biomass compared to the peripheral blood, but not to other organs. So the biomass in the spleen is not uniquely high in these patients compared to all the other organs and compared to the blood. And finally, we didn't find any gametocytes and no evidence of sexually committed cells in the spleen in this cohort. So, but of course, we did find that in the bone marrow, which provided us with first direct evidence that gametocyte formation actually happens in the bone marrow. So with that, of course, we were somehow conflicted because we were already involved in the other study with Stephen Coe. We saw all the data. We helped them with the quantification of the histology, so it's not wrong. But what does it mean? So at the moment, we are in discussions with them, and what we concluded for the moment is that we basically look at two extremely different sides of the aspects of a gradual phenomenon. So on one hand, we have the asymptomatic chronic adults, for example, those in West Papua, with a long duration of infection, a lot of exposure, probably chronic anemia, and subsequent and constant reticulocyte production, and potentially the establishment of a tolerogenic environment for the parasite in the spleen. On the other hand, in Malawi, we have this pediatric cohort of severe fatal malaria in children with very strong activation of immune responses, a very inflammatory environment, and regular treatments. And so at the moment, we have nothing that somehow matches both findings at the same time in a cohort which would probably be one of uncomplicated malaria, which is yet to be established. So we have a few cases of incidental parasitemia in Malawi, which we are currently looking at. But otherwise, this is the hypothesis that we have been coming up with, and that's probably one of the explanations for these seemingly incompatible findings. And so just to recapitulate, of course, the bone marrow has specific functions, including hematopoiesis, immune cell production and maturation, storage of stem cells, release of red blood cells. And the spleen plays a very complex role as well, in particular filtering of infected red blood cells and old red blood cells, production of antibodies, activation of immune cells. And we want to now take a step further. You have seen a glimpse of this already by João and also Fabio's presentations. And really, the idea is to develop a cell atlas of bone marrow and spleen in malaria infection using spatial omics, and then validate these findings using high resolution or deep imaging and ex vivo and in vitro experiments. So this is what we have been embarking in the last 5 or 6 years. On one hand, of course, with our friends in Brazil, in particular Fabio and Marcos Lacerda, Welton and Giseli, and then, of course, also with collaborators in Glasgow. This is Thomas Otto and Kevin Cooper, and talented people in the labs, João in particular and Jessica for a study in Brazil, and then again João and a Malawian PhD student Simon for a companion study in Malawi, where we basically look at this cohort again. And the idea is essentially to look at the historical cohorts that we have, both in Brazil and in Malawi, but also at prospective studies, in particular longitudinal ones, like Fabio mentioned before, and then perform a combination of imaging mass cytometry, where we have a multiplexed antibody staining, spatial transcriptomics, and then also a suspension single-cell RNA-seq to complement it. So we got 2 grants that we are working on. We were a bit derailed early on because of COVID. On the other hand, it allowed us to basically apply the planned tools to lung cohorts of fatal COVID in Brazil and in Malawi, and basically already test our multiplexed imaging mass cytometry approach in both cohorts, and also single-cell RNA-seq from the infected lung in the Malawian cohort, and then compare the cohorts, for example, and also compare them to American cohorts, from which, of course, there were many more data. So we published these 2 studies, and then eventually were able to set up the actual experiments with falciparum and vivax. So here's Joao and then Simon, the PhD student. This is for the falciparum data in Malawi. And we have seen these before now, but essentially you can make these UMAP projections of the antibody responses, and you can identify cell types based on the particular markers that we use, and you can then look at the cellular distribution of these, or the cellular distribution across different patients or patient types. So we have non-malarial controls in Malawi, we have incidental parasitemia. These two, of course, are very few because they were misclassified originally. And then we have these so-called CM1, 2, and 3. So CM1 and 2 have cerebral malaria. CM1 and 2 probably both died of it, but CM2 also had a lot of vascular damage and ring hemorrhages. And then CM3 had less cerebral malaria and probably died of other diseases, like, for example, meningitis. So we have these 20 patients that we are working on, still the original set from Regina Joyce and then from Lauren, plus a few more. The next steps, of course, are now this systematic analysis using imaging mass cytometry and cosmics, or spatial transcriptomics, validation by IFA and IHC, and ongoing complementary longitudinal experiments. We also want to embark in larger cohort studies and include India for a comparison with Brazil. We are also very interested to start looking at organoid proxies for the autopsies that we have. And one interesting aspect is we tried to predict blood biomarkers, or to use blood biomarkers to predict bone marrow infection, something that we have been discussing in particular with Fabio since quite some time. So with that, I want to end. So there was a whole bunch of people involved over the years from both Glasgow and Zurich more recently, but in particular, of course, Regina Joyce, Lauren Galloway, and then Joao. And there's lots of collaborators, our friends in Brazil, our colleagues in Malawi, a number of other ones, and we were very lucky that we could get this funded, and now we try to renew it. Thank you very much.
Thank you, Matthias. And we continue now with Dr. Sylvie Garcia. She's a researcher at Pasteur Institute in France, specializing in host pathogen interactions in immunology. Her work focuses on humanized mouse models to study immunoresponse and infectious disease. Please, Sylvie.
You hear me? Yeah. Do you have the pointer? Okay. So first of all, I would like to thank a lot Daniel and Carol for their kind invitation to this wonderful meeting. I'm very happy to be here. It's my first time in Brazil, and really enjoy it. So thank you for the invitation. And I will present you some data on our work on Plasmodium vivax and bone marrow infection. So it's a bit difficult to go into this slide after the presentation of Fabio and Matt, but what we well, I think I will show like this. As the previous people told you, is that more or less the spleen is a reservoir of asexual stages, I think, at least earlier, and responsible probably for chronicity, while the bone marrow is still the sexual reservoir for P-Vivax. So in this study, so let me see if I can do something like this. Well, it's very difficult. So we concentrate our study on the bone marrow reservoir. Okay, so our question were basically, what are the host-parasite interaction within the bone marrow niche during Plasmodium vivax infection? And we wanted to look at that in vivo, and of course to do that in vivo, it's difficult to have access to bone marrow, at least in our case. So what we did was to establish a humanized mouse model to study host-parasite bone marrow interactions. So first, during my talk, I will start by showing you our results on the new humanized mouse model. We called his hairy mice for human immune system, human erythrocytes. And then, so this is published, and then I will show you some new data we obtained on host parasite bone marrow interactions. Okay, so what we did was designed a new host, immunodeficient host. So I will not detail everything, otherwise I will be late, but if you want to have more details, you can come to me after to discuss. So this type of model, they consist in engrafting some human hematopoietic progenitors, which are able to give rise to all the white cells and red cells from the blood, into immunodeficient mice. So in our new host, the classical model were very poor at producing red blood cells, but we managed to do it. And here, so I'll show you here, you can see that this is blood flow cytometry study. Here is the marker of murine erythropoiesis. This is glycophorin A, so for human erythropoiesis, and you can appreciate the human red blood cells which are produced in our bone marrow mice. So we have about 25% of the total red blood cells within the bone marrow which are from human origin. And as you can see, this is CD235A, so glycophorin A, human CD71, and we have all the stages of this erythroid human development. Here is another way to follow the development using a double staining with CD49D and band3, and we see the typical waterfall profile that we expect in human bone marrow. So in this model, the vast majority of the cells express CD71 positive, CD71 marker, but also the dark marker and other marker useful for vivax infection first and typical for human erythropoiesis. So of course, our question was if these hairy mice were able to be infected by Plasmodium vivax. So is what we did in collaboration with Marcello, with Plasmodium vivax isolate that we inject IV into these mice, reconstituted mice, that is they have some red blood cells, human red blood cells inside. And as you can see, this is the bone marrow. So this is the number of copy of cytochrome B RNA for 10 to the 5 human red blood cells, and we follow the infection at 1 week, 2 weeks, and 3 weeks, but we can also see that after 2 months of infection, we see that the Plasmodium vivax can be maintained in these mice. Second, we ask whether this bone marrow, infected bone marrow, could be transferred, and infect a secondary host, right? Non-infected mice. And as you can see, so this is the parasitemia of the first infected mice, and when we transfer to a secondary host which is naive, you can see that they can also be infected. So this is all in the bone marrow. Oops. So then we ask if the cells, infected cells, we were seeing within the bone marrow were from the isolate we injected or de novo infected, newly infected. What we did is that we use a trick. That is, we injected some rhesus D negative infected cells into humanized mice reconstituted with cord blood, which were rhesus D positive cells, so that we could differentiate the red blood cells produced by the mice from the red blood cells infected from the isolates. Okay, yeah. So to do that, of course, we had to use flow cytometry, and to detect the infected cells, we adapted a method, the Stellaris method, to be able to detect by fax the mRNA from the parasites. So we call it FlowFish. It's published if you are interested. So what is here? This is the human red blood cells marker. This is the 18S mRNA from the parasite. So this is here in this quarter is infected cells. So we ask whether these infected cells were expressing rhesus D or not. And as you can see, by 1 week after infection, but we show it even before, I think day 5 it happens already, all the cells which were infected at this time in the bone marrow were newly infected. Okay, and what we did is that we process these cells through image stream approaches, which is a combination of both cytometry and microscopic analysis. And as you can see, so this is a marker for human red blood cells, so this is the bone marrow, so this is basically the cells. Are the cells which are de novo infected. As you can appreciate, this is a DABI staining, they are nucleated, and they are also infected. So what we detected in this bone marrow is nucleated infected cells. And of course, the reviewer asked if we were able to see infected cells by GMSA, but they were nucleated cells, so I tell you it was a nightmare. But my wonderful student at the moment, she was raised in the field by Marcello, so she was very good at GMSA, and she could manage to see some rings, and even she told me some trough. I believed her, so, and the reviewer also, so it was fine. Okay. It doesn't want to, okay. So then, we asked whether the bone marrow were able to support sexual differentiation of the parasites. Oops. So again, we use the flowfish method to detect some gametocytes since, I mean, if they were nucleated cells, it would have been very difficult to check, and it was. So this is, again, gated on human cells, so CD235A positive cells. And here is the 18S mRNA, so on that column is infected cells, and we ask if they were expressing PVS25 mRNA. So I don't know if it's the right marker, because I hear that it may be not, but I mean, it's the way we did. And as you can appreciate, we had here 1 in 4 cells which were sexually differentiated, and much more 2/3 differentiated cells within the bone marrow. So we conclude that, so we conclude that the bone marrow were indeed a reservoir for sexual stages, because we found much more sexual stages in terms of proportion in the bone marrow than in the peripheral blood. And again, we look at in the flowfish method, and as you can see, in the blood it was very difficult to detect the gametocytes, but in the bone marrow you can see some PVS25 mRNA expression within nucleated cells, and you can see the 18S marker. Okay, so then of course the next question were if these gametocytes were transmitted to the Anopheles. So we made our poor mice bitten by mosquitoes, and they did it very nicely. I cannot tell that, but there were plenty of mosquito under the belly of our mice. And 6 days after, we look at in the midgut, and I don't know why it doesn't work so much. Okay, and we find some oocysts. Not so much, but we find some. Okay. Tell me. Okay. So I will go there. Okay, and then 2 weeks after, we look at in the salivary glands to see if we could find some sporozoites. And the way we did that was to use image stream also, and you can see the shape of the sporozoites. This is a staining with PV-CSPs in green, and here is the epinucleus. So we were able to see some sporozoites. Okay. So this work was done with a wonderful student, Camilla Luisa Battista, who was an ex-M2 from Marcello's lab, and we found that we think that we established a new humanized model for long-term Plasmodium vivax infection, and we allowed to maintain the parasites through bone marrow transfer. And within a week, and certainly before, all infected cells were from de novo infection. And we think that this model reproduced several features of natural infection. First, the predominance of the gametocytes within this organ, and second, the infection of nucleates itself. So that I don't know, we can discuss it. This is what I believe. And the transmission in low parasitemia settings, because we found that even though some mice had undetectable parasitemia by qRT-PCR, they were still able to transmit. So I think it's a sensitive model for transmission. Okay, so then I go too quick. So then we decided to study the early bone marrow Plasmodium vivax interactions in the bone marrow. So what we decided to do is to restrict our analysis before the onset of symptoms, to see how the parasite could establish before the peak of parasitemia in the blood, as it was described in, say, HMI from Simon Draper, for example. So before 2 weeks and before this systemic inflammation. So, so what we did is that we studied this interaction at the level of immune system first, immune cells, at the level of erythroid cells, and we are doing at the moment at the level of parasites. So I will show you example of the first two ones. So what we did was to use single-cell RNA-seq, because I'm immunologist, but I didn't know which cells I could see. There was too much. So what we did was to sort the human leukocytes from the bone marrow of infected mice. We subjected them to 10x genomics, and as you can see, for the UMAP blot, we were able to see the all type of cell subset we expected from the human bone marrow. Okay, and what we saw, looking at the different population, we saw a very rapid monocyte polarization switch in the infected bone marrow. And this switch was prone to be anti-inflammatory. So you can appreciate that from the total mature monocytes, from the differential gene expression analysis. And what we did was we subcluster the monocytes, the mature monocytes, into different populations. Most importantly, the classical CD4-1, the so-called classical and the non-classical cells. And we look at day 3, day 7, and day 11 in both classical, non-classical cells. And as you can see, quite early, we have a drastic downregulation of the type 1 interferon signature, ISG, and we have also a downregulation of the inflammation response that both goes up later at day 11 after infection. And this is correlated, so the downregulation of type 1 signature is correlated with a higher signature of anti-inflammatory genes. So this anti-inflammatory signature goes down about day 11 after infection, but still, you have still some anti-inflammatory gene expression, right? And this goes together with the downregulation of glycolysis metabolism, which is specific for inflammatory monocytes. And as you can see, this goes up again at day 11. Okay, at the level of erythropoiesis, what we found, so again, we sorted the human red blood cells from the bone marrow, we applied 10x genomics, and then as you can appreciate, we have a very high impact of the P-Vivax infection on this process. However, at least in this is day 7 post-infection, we didn't see any obvious changes in the erythropoiesis, except for the level of hemoglobin genes, which went down very drastically. You can see that also at the level of the proteins. Okay. And what was striking also is that some cells express a high signature of type 1 interferon. So you can see that through through some expression of some genes, which are EFITIM3, for example, or E56 or ESG15, and it goes together with a high expression of class 1 and immunoproteasome gene, associated gene, and a lower expression of hemoglobin. And we found it again with here, right, with upregulation of HLA-A2 and upregulation also of FITIM3. I'm not very good at that. Okay. So this was done with, also, I'm very lucky with my student, a very good student, Dorena Schul, and I think that she showed quite well that P-Vivax induce early immune response in the bone marrow, that there is a rapid modulation of early interferon-mediated response, and a early shift in M2 anti-inflammatory monocytes, which afterwards later is modulated and is less anti-inflammatory, but it's a bit, again, more inflammatory. This is associated, I didn't have time to show that, but with CD4 and CD8 T cell suppression. And also, at later time point, also day 11, the starting of emergency myelopoiesis. And what we show in the erythroid cells, we had for some type 1 responses, so I don't know if it's associated with the prevention of Plasmodium vivax infection, since it's not a virus. I don't know if it's efficient, and is associated with the downregulation of hemoglobin genes. So we believe that it's at this early time point after infection, we have a kind of trade-off between parasite clearance by the immune system in the bone marrow and its maintenance in the bone marrow. So I have 1 minute, so it's okay. So what we believe, if I want to do a scheme to recapitulate what we
Então, para recapitular aqui o que achamos que ocorreu, ao injetar o parasito, vimos o interferon tipo 1 e um pouco de tipo 2, que é mediado pelo próprio parasito, e a resposta imunológica humana a esse patógeno. Vemos aqui algumas outras respostas, mas com certeza há uma resposta imunológica tipo 1. Essa tipo 1 seria o compartimento de células imunológicas, que fisicamente, geograficamente poderia ser igual, e também a ilha de eritroides, onde as células se desenvolvem, e os macrófagos cercando as células de eritroides em desenvolvimento. Então, no início, podemos ver uma resposta inflamatória, não foi identificada aqui, mas identificamos a intermediária, M1 e M2 ao mesmo tempo. E, como eu falei, temos um fenótipo M2 bastante claro, o que é interessante. Há muitos genes que também são expressados durante o câncer, no tumor, no ambiente cancerígeno de alguns cânceres. E, num ponto mais tardio, ainda vemos uma expressão de genes inflamatórios M2, mas também um aumento na resposta de interferon tipo 1. Isso está associado, com certeza, com a supressão de ativação de células tipo 1. Não aparece tanto assim, mas são células suprimidas. É diferente. E, ao mesmo tempo, vemos a resposta tipo 1 aqui, num compartimento diferente. Neste caso, talvez possamos ver a resposta tipo 1, mas já que o parasito pode se replicar novamente, acreditamos que depois vemos o mesmo sinal e uma nova onda de resposta interferon. É um equilíbrio entre o controle e a evasão humana. o que faria parte do controle da medula e do parasita é um pouco difícil de discernir. O nicho da medula precisa ser protegido, e haveria uma barganha necessária. Não encontramos uma parasitemia maior do que 3%. É bastante contido. Aqui é a minha equipe. O primeiro estudo com camundongos foi realizado pela Camila. A interação foi feita por Duranda. Será publicado em breve. A Liliana Mancho e a Eliana Real também ajudaram bastante com a análise. Arthur Schaff também é um estimado membro da equipe. Daniele, Silvia e todas as instalações que auxiliaram. Muito obrigado.
Thank you for all the speakers. Now we invite you, Fabio and Matthias, please, to the stage. And we are open for questions. I think we have like a few minutes for a few questions.
I'll get started. Thanks for the amazing session. So Fabio described the increase in precursor erythroid, but the reduction in the mature forms, it sounds similar to the dyserythropoiesis of falciparum. So I'm wondering what the features are that might distinguish what's going on with falciparum versus Vivax, and specifically what's happening to the red cells. Are they being destroyed, like what Junqueira described with CD8 T cells? Is there an arrest because of the immunomodulation that Sylvie's describing? Or is it just early release, which happens with anemias, that there's an early release of less mature reticulocytes? So what's going on with the red cells?
Well, we still try to understand what's going on with the Vivax for the bone marrow. Falciparum is quite different, right? You have lots of rings. So I'm really not sure. I believe the way that they behave in the spleen in the falciparum and Vivax malaria is quite different as well in the blood marrow. So we see lots of glomerulocytes in the blood marrow, and the spleen in the falciparum malaria is not complete, it's different from the Vivax malaria. Mm-hmm. So we have lots of asexual blood forms and the spleen in the Vivax malaria. So the spleen Vivax malaria, in my opinion, plays a huge role, much more important in terms of the resistance for the host.
Okay, and then about the red cells. What happens to the red cells that are maturing? Where do they go to? You said there's an increase in precursors, but there's a reduction in the mature forms. Yeah. Do we know where they go?
No, not yet.
Do you know? No, we don't know. Sylvie? Thank you.
Fantastic session. I love the three talks. So I have questions for all of you, but I'll start with Sylvie. What's the time period that you think between the inflammatory and anti-inflammatory response that you're seeing, or that you predict to have with Vivax?
That is the time between inflammatory and anti-inflammatory.
So high interferon and low.
I think it's 7 days. In 7 days we have. It's a very rapid conversion.
Okay, thank you.
So who is doing that? We don't know, but it's quite rapid. It may be the bone marrow.
And then for Fabio, you mentioned that there's like two Vivax, one in the peripheral blood and one in the bone marrow spleen. Did you correct for cycle in the single cell analysis? So could it be that it's some early stages in the peripheral?
Well, that's a very great question. I was talking to Matthias yesterday about that. We still do not know, but maybe that different, okay? But still have to try to find a way how to analyze that data, okay?
Yeah.
So what's that? Well, is another way, okay?
Maybe you could use the great tool from Anna. Now that she could generate it, that would be great. And then for you, Matt, more speculative? You're aiming to have like this side-by-side comparison between vivax and falciparum in the bone marrow. How would you speculate the immune response or host response would be in this medium spot?
You mean in the uncomplicated cases?
In both.
Yeah, we just, I think the problem is we only have systemic measures at the moment, so we don't have any data from organs. And as we already know from the studies that were done in Vivax now between bone marrow and blood, day 0 and 42 or 60, that what we measure in the blood is extremely minor compared to what we measure in the marrow, so we can't really tell. It would be really important to have a cohort that is specifically targeted to that, which would have to be some kind of a comorbidity cohort, because obviously it's ethically very difficult to get sampling if there's no reason for it.
Yeah, thank you.
Yes, very good session. I congratulate you all. My question is to Matthias. For how long do you have to wait from the time of death of children and the collection of organs and samples from the corpses? I'm asking this because gametocytes, for instance, they are produced in vitro culture when the stress appears. So when you have a culture that's not going so well, the gametocytes can appear. Do you think that something like this could arise also for a long time after death? And would you be able to collect organs or samples at different times after the time of death?
Yes, thanks. That's a good question. It was actually a reviewer comment for the original study too. So the post-mortem interval, we only analyze patients that have a post-mortem interval below 12 hours. And the reason originally was just that the tissue quality was not sufficient otherwise to do histology, in particular immunohistochemistry. But then, so if, let's say, within these 12 hours there would be some kind of stress response and sexual commitment, obviously that wouldn't be enough time for gametocytes to develop. And so, given that we see a similar proportion of sexually committed chysons with MSRP1 and gametocytes with PFS16, I think it's not possible that this would be due entirely to that phenomenon, because the ratios are similar. Yeah.
Or migration from organ to circulation in the vessels instead of bone marrow.
Of course, we cannot exclude that because the parasite dies not at the same time as the patient dies, that there was some post-mortem activities that we have not captured. Although, of course, by average, there's an 8-hour delay between transcription and protein, and so that would be a very narrow window where something would happen. But we cannot exclude it. That's absolutely true.
A question for Fabio on the Vivax single cell data from the bone marrow. That you talked about sexually committed cells, and you showed AP2G, I think, higher expression. How else you define that this is a sexual commitment in Vivax? So do you see other markers like in falciparum PFS16 or any other of those, or how do you do the definition?
Well, that's one thing that's really, let's say, not easy to analyze. We always use the markers based on falciparum malaria, and I'm not sure if it would be that's a proxy, right? And of course, the one thing we see sexual commitment in rings, troughs, gamerocytes, but the question is, and still, and you might have to talk this later on in a coffee break. What this in terms of, sex commitment is something, to be gamerocytes is another thing. And we're really not sure by this time if we have a very good market to show this. So this we could talk about later, but this is a very key question that's quite interesting.
Just one more, Fabio. In experimental models, for how long do you find parasites in blood marrow?
Well, no, that studies we just found on day 1, okay? The convalescent patient, that's day 42 and day 6, will have no parasite in the bone marrow. But that's one, this is a very good question, Claudio. Because, and that's why I was talking to Matthias, we talk about blood aspirates, right, in the bone marrow. And we have a limitation of the blood aspirates. Yeah. I'm not sure if when you take this, the collection of this blood, you have everything that's in the bone marrow, right? And I believe that the answer is very, very good question. I believe that the answer comes from the biopsies and maybe in the mice model, okay? Because we have all this limitation. Okay, the aspirates will just not go that deeper and so on. So that will come from the biopsy that João is going to do the analysis for the biopsy for Marcus and probably in the mice model. So let's see the next meeting. Maybe I can answer you, okay? Okay.
Yeah.
I think I have time for a last question. May I ask you a question, Matthias? I don't know if I understood correctly, but the two studies are not maybe directly comparable because in the autopsy study you had the spleen and other organ samples, but you did not have the blood parasitemia, or did you have that? Because I didn't see the...
So we, at autopsy, you cannot take blood, obviously, because there's no blood pressure, so we don't have blood samples from most of these cases. But for that reason, we used the lung. So we basically quantified parasitemia in the lung, in the vessels, based on immunohistochemistry, and used that as a proxy for peripheral parasitemia, which, I mean, it's the best we can do, using large vessels for that.
But when the patients got there, did you get the parasitemia, the blood parasitemia, when they get before treatment, I mean?
Yes, so we have that, and that is usually similar to the parasitemia that we calculated in the lung. So we have several parasitemia measurements when they came to the ward and before they died. That's correct, yes. But we wanted to use the one at the time of death because it's the most accurate proxy.
But sometimes they have been treated for long hours, right?
It's a good point. I mean, we could probably plot both. That's good. That's right.
Okay, thank you very much.